All right, okay. Everyone can hear me? Great. Hey, before we get started, as people start coming in, I just want to get a feel for the room. How many people are here who are from like a life sciences, pharma background, biotech? Okay, so a few of you. The first half of my talk, I'm going to be focusing a little bit about drug discovery, how the process works, so bear with me. It might be a little boring for the rest of you. It might be new info. How many of you do have a data science or data background? Great, a lot of you. Great. Perfect. How many of you is the first time Spark Summit attendees? Wow. All right, welcome. I gave a similar talk in the last one at San Francisco, but there's new stuff if you were there. Anyway, all right, so a little bit about me. My name is Vishnu. I'm the founder and CTO of WiseCube AI. We're a life sciences AI company. I'm not a molecular biologist or a medicinal chemist or any of that. My background has been mostly in software, building platforms, e-commerce platforms, machine learning platforms, and so when I decided to do a startup, I was like, all right, I'm going to build a platform because that's the only thing I know how to do. But in the last year or so, we've been closely working with a nonprofit biotech firm called IDRI to help them sort of accelerate drug discovery using machine learning and AI techniques. So it's been a great learning experience for me and my whole team, and also it also made me realize like I should have paid more attention in a high school chemistry class, because there was a lot of chemistry that I was like I'd forgotten that. Anyway, so just the agenda for today, we'll go through like the history of drug discovery and drug screening and molecular representation and all that good stuff, just to get like hopefully 80% of the audience caught up. And then once you become experts in drug discovery process, then we'll go into the actual case study. I'll show a quick demo of some of the pipelines we built, and then we'll take questions. Sound good? By the way, if you do have questions, like please raise your hand. I want to make it as interactive as possible. Don't have to wait for the end of the session. Always ask me questions in the middle too. Just raise your hand and we'll stop and do that. All right. Let's see if we can breeze through this history of drug discovery. So we know there's always been like diseases ever since there's been humans, and we've always been trying to solve it. And the way we've been trying to do it has been varied. Like this example, the Chinese medicine, Ayurveda has been around for thousands of years. It's a fun fact. How many of you know that I actually found this out recently? There's a difference between inoculation and vaccination. Do any of you know what the difference is? It's a subtle difference, but it's sort of like, I was like, what? I didn't even know that. So I'll give you the answer. The inoculation, apparently, it's like you take the actual, the scab or like the virus itself from an infected person and you apply it on your skin. And then vaccination is sort of a slight variant. The difference is it's like a variation of that virus, which is not directly that virus, but it's a variant of that virus, hopefully a more harmless one that you then inject yourself, which doesn't kill you. Anyway, so people have trying to do this for thousands of years. More recent efforts, of course, the cowpox was the example of smallpox. Vaccines. People found out that if you get cowpox, you don't die of smallpox, and so things like that. Penicillin, obviously, the drug. And that caused like a whole explosion of other medications to come to being and so to this point, like the analogy I sort of use is like people were trying to find cures without really understanding how the mechanism actually worked, right? So this is why this process of I'm going the wrong direction. The process of rational drug discovery was, I think, a critical moment where it was to me at least, it's like, hey, we've got a lock. We don't know really what opens the lock, so we're just going to try every key we can find our hands on till one of it actually works. That was the process till this point where we call rational drug discovery, which is effectively saying, look, we're just going to understand the lock, try to see what the lock actually looks like before we start trying out keys. And that seemed to be more effective, as you can imagine, than just trying every random key that we get our hands on. So when we started narrowing down in terms of how they actually came up with the keys, so to speak, the drug targets, actually in this case, these drug targets would be the locks. Would be like phenotypic screening, gene association studies, imaging, biomarkers, which are basically looking for things that the body will react to for a certain pathogen. Like it's not directly the pathogen, but there's like other markers that are like, reliable indicators that the pathogen exists or is inhibiting, either case, right? Let's see. This is you would see this, if you're in the pharma industry, you probably know this by heart, like where, hey, the target, and you do the validation, and then you go through the lead discovery phase, and then now you're trying to optimize the lead. We'll talk about this more in other diagrams, and then you finally go to preclinical testing and clinical testing and finally get to market, right? So that's sort of the pipeline. Everyone talks about this pipeline, right? So you heard this a lot. And the screening methods have gotten a lot better. They have these robotic arms that go and do stuff, high throughput screening. They can do thousands of potential targets. It's gotten really, really fast to be able to screen things. But at the same time, it's still a very, very much a hit or miss process, right? So you try to screen thousands of things, and you might get like a few hits. And then once you get those few hits, you try to optimize these into leads, and then you get even less. So we'll talk about this, where this whole process of hit to lead optimization is still very error prone. It's still a problem where finding leads and finding things that actually turn into drugs that can start the testing process is so error prone, and it's because you start with thousands at the top, and I'll show you this funnel, and by the time you get to the phase of preclinical testing, you now have a handful of things you can actually even start testing, right? And we'll talk about all the differences between ligand-based and structure-based things too. But once again, this whole thing where you have a collection of compounds, you run some assays or tests, and then you see which ones actually can turn into leads, and then you try to optimize those leads, and then you say, okay, which of these leads can actually be turned into drugs or drug-like things? This thing called drug-likeness, you'll hear this a lot, which really means which of the compounds that we've identified that we think could have an impact on the pathogen or whatever it is that we're trying to have an impact on can actually be turned into a drug, so that one, we can administer it into the human body when it actually can be metabolized, and then also can be then moved out of the body. And so you'll hear this criteria: absorption, right? Distribution, metabolization, excretion, and so the ADMAKE criteria. So how can you make a stable enough drug that can survive the human body but still be effective, and then finally get out of the system as well. So anyway, so there's lots of criteria that goes into this stuff. How many of you seen this particular graph? Eroom's law, great. So how many of you heard of Moore's law? All right. So Eroom's law is just the opposite of Moore's law. Literally, it is the opposite. If you write more, in the opposite is Eroom's law. So there was a paper, a Nature article that was, I don't know what the year is. It was definitely 10 years ago, where they talked about the drug industry overall, and there's sort of a disturbing trend in how new drugs came out. And it was actually, they found out, even though with the high throughput screening and all of these other techniques I spoke about, the number of new drugs that were coming to market were actually reducing over time. It was taking more time and money to put out drugs, and so that was the trend line. They said and of course they attributed to various different factors. If you read the paper, it talks about FDA tightening regulation because of the thalidomide incident in the 70s. And then there was just a backlog of things, and new therapies coming in and all of this stuff, but the trend remains, right? You can see the trend, it's very clear. It's downward trajectory. The number of drugs keep going down per $1 billion spent in R&D. And so now it takes decades and billions of dollars to get a new drug out, which is, it's not that diseases are going away. We have more diseases now than ever before. Yes, we've solved a few, but it's still there, and there's more people. So this is a very disturbing trend if you just hadn't thought about this, right? And this is where obviously we're here today to talk about how we think data and machine learning and AI and all these other techniques can potentially, at least correct the trend and hopefully reverse it. right? So we'll talk about the timeline once again. So this is sort of the different stages of a drug discovery timeline. In stage one, you do like the true discovery R&D phase. You start out with tens of thousands of potential compounds, and then you start like screening them and start weeding them out to the point where like, hey, these things have an effect. These things can actually be drug-like, and then you get down, and that takes years, right? And you get down to stage two, where now you start to do like sort of preclinical testing on animals and make sure it's safe enough, it's not too toxic, it can actually be made into a drug, all those things. And now you're left with like from 10,000, you're left with like a few hundred. and that takes, and then by the time you get to the clinical stage, it's less than like a handful, right? And so to get that one compound that actually clears, FDA approval and clinical testing, you have to start with the tens of thousands, literally at the top of the funnel. So the goal, at least, there are companies that are trying to solve this, all these different stages, right, and using various techniques. At least, our focus has been mostly in the sort of the first stage, the top of the funnel, where you start with potential compounds and you're saying like, how can you narrow down the right set of compounds so you'll have more left when you get to stage two, right? So that's sort of our focus. And the way you do it, traditionally, there has been two sort of broad camps of like what they call computer-aided drug design, which is one that looks at the structure itself. When the structure in this case is the structure of the actual protein or whatever it is that you're trying to actually latch onto, right? And then the target structure. And then the other way is the ligand structure, which basically means it's the key, right? So you look at going back to our key analogy, is if you know that a certain key actually works, then what you start doing is you start looking for keys that look similar to the key that works. That's sort of the ligand-based approach, which is like, hey, I know this ligand structure actually works for me, and I actually went through it. So I'm going to start looking for similar keys by looking at like different things and doing virtual screening and so on. So that's sort of the approach that we also took with the case study I'll talk about. It's more about like, can we start with a set of compounds that we know work, and then like figure out, how can we predict ones that we've never seen before, or potentially come up with new ones, right? And then you get to the lead optimization and the candidate stage. So now we get into like the actual problem of what we're trying to do. Is the biggest problem we need to solve is like take a thing that looks like this and sort of represent it in a digital format, so we can actually start doing some analysis on it, right? And ultimately, that's what we want to do. And that's sort of, as you can imagine, like this thing is a pretty complex graph of like things, elements, and different bonds, and like there's all this. Like, how do you represent this in sort of an efficient digital way, right? And there's different ways people have come up with over the years. Descriptors, if you will. You just look at it a one-dimensional way, where it's just like, I'm just going to look for things I know about this compound. Like, I know its molecular weight, what is its solubility in water, maybe its charge, the number of bonds it has. All of these things are one-dimensional things, or scalar values that I can calculate, and I'm going to use that as descriptors, right? And there's like simple rules, like Lipinski's rule of 5, or Pfizer's rule, where then you say, like, if it has these things, or if it doesn't have these things, then it's probably okay. And then you start like weeding out, which compounds to go after, which are not good ones, and just based on these sort of scalar values and descriptors, properties that these compounds have. And they work to a certain extent, which is like, it's a good way to weed out things. But remember, the molecular space itself is just huge. I think the number of potential compounds there are is in the order of like 10 to the power 60 or something. So we're talking about a really, really large space that we're trying to sort of navigate to find out potential hits, right? And so therein lies the challenge, where like, how do we be more effective around that? It's really a search problem if you think about it, right? So the other way you can like describe these molecules is a very common way. This is the 2D descriptors, or circular fingerprints. And then the intuition behind this is you look at a compound structure like that, and you sort of look in 2 dimension, like what are the other sort of bonds or elements around any given chemical element, and then you start, coming up with sort of vectors or bits to represent the things you start finding around it. So in this example, for example, you start with like one of the molecules there, and you say like if I have a radius of, and you can define what the radius is. This is an example of what they call the ECFP, which has, and you can say ECFP4, which, for a radius of 2, then you say, okay, around 2 bonds around my molecule or a chemical of interest, I'm going to see what other things exist, and then I'm going to start like, putting out like bit vectors if there are things around me. And then you start doing that with one end, and then you walk your way through the whole graph. And then at the end of it, hopefully you'll have a bit vector with, with a bunch of ones and zeros. And that's sort of your fingerprint, if you will, for your compound. And there's different variations of this, but the radius differs and where you start and all that things. But effectively, this is a very well used way to sort of featureize your compound, if you will. Right? Yes.
Thanks. So this description gives him some features of the properties of the molecule, but is it also like a full description? Could you, from the bit vector, then reconstruct the molecule, or is it not one-to-one?
Are you saying can you go from a bit vector to a molecule?
Yes.
No, I don't think it's almost like a hash. If you think about it, it's a one-way hash that you come from the molecule to the bit vector, because potentially multiple molecules could have like similar hashes.
Yes, so it's not unique.
It's not unique,
okay,
right?
Thanks.
And that, by the way, that's a great problem where like you could have molecules that are different, could potentially have very similar fingerprints, and vice versa. So it's like, and it gets even worse because chemically they might actually be very, very similar. There might be a compound that actually looks exactly like the one there, except I replace the oxygen molecule with something else, right? And the challenge is, from a chemical sort of looking at the structure perspective, they're very similar structurally. But the biological effect it could have on a human body could be like very, very different. And so that's another big challenge in this whole space. It's just chemical similarity does not mean biological sort of impact similarity, right? But we do our best with what we have. This actually works, and then there's other ways you can also you can do fingerprints around. I'll talk a little bit more about like neural networks and how they can use autoencoders for fingerprinting. But once you have these, then you can actually start doing things now, because now we know, okay, once we got a fingerprint, it's a bit vector now, and we know what to do with bit vectors, right? We know once we featureized it, we can do all our favorite algorithms and do all of that stuff, which is great. And then you start doing things like this. Hey, I take the data set, I have the descriptors, and by the way, you don't have to stop with just a fingerprinting. I'll talk about like in our case study what we did, but like you can use the fingerprinting as just one feature set, right? So you can add other things like properties of the compound you found, the number of bonds, and other things that you find about the chemical compound, and then just sort of enrich that your feature vector, if you will. You don't have to restrict it to just the fingerprint. And then you can start doing things like this. You'll hear this term QSAR, which is just a fancy way of saying like, hey, I'm going to use the structure of the compound to find out how it's related and what is the activity the compound has on specific things, and then being able to quantify that, which basically is just using statistical techniques and machine learning things. I guess they don't want to use machine learning or AI, and so they call this weird QSAR thing, whatever. But all that means they're using statistical techniques to be able to predict things about the compound itself, right? This is the other biggest, if you learn nothing else about this talk, if you learn what a smile string is, you probably know more than 90% of other data scientists in this field, this is the way to represent compounds in a sort of a textual format. So you can, and this is like, it's the industry standard way. So you take any chemical compound, and then you can turn that into literally an ASCII string. And so in that case, you start out with one end, and then you say like, what are my, compounds or elements I find, and what are the bonds that it has with the next element, and then like you start writing that down, and then you get a long string like that. And that means like, hey, there's a nitrogen, it has like a single bond with this other carbon, it has another carbon and another nitrogen, and so on. And there's a way, and then he's like, oh, there's like a, and then the bond like this, and then you write that. And now the problem is there are different ways you can write small strings, and like depending on which end you start with, it might actually be different. And And so they have another way to like sort of canonicalize this and normalize it so it's the same no matter where you start. But this is the way to represent compounds. And typically, when you're dealing with data sets in this space, you're dealing with things that look like that. And so so like that's what we did. I'll talk about where we had like 100,000 of small strings that look similar to that. And so, of course, the ECF4 or whatever is not the only way to do fingerprints. Now, of course, with deep learning, you can do neural fingerprints. You can actually run it through an autoencoder and try to get the latent representation of the graph itself, the molecular graph. And there's really cool work being done there. I'll talk about some papers where they have a graph neural network, a graph GCN convolution network that can actually be used to represent the whole molecule as a graph, and then being able to do things with it, and it has some very interesting results. So this space is pretty hot right now, but so yeah, we're watching this to see if like this can be actually really useful for some of the things we're doing too. So taking a little step back, so we talked about that whole funnel. One of the things that one of the reasons at least that drugs like fail to make the phase 3 or all the way to the end of the funnel, and they realize, oh, there's something wrong, you got to start over. You might have spent billions of dollars in a decade working on this, but sorry, it doesn't really meet, and FDA can like actually throw out the drug in phase 3 trials, which means now they're doing it against humans, but they're doing it like not just for safety, like the efficacy, and then also making sure it actually works like when people are taking other drugs and all the other stuff. But the number one reason, as you can see in this chart, is like efficacy, which means that it actually doesn't do what it's supposed to do. Right? That's the number one reason that the phase 3 trials actually fail. The number 2 is safety, right? And there's number 3 is commercial reasons, which is just like the pharma company runs out of money and we just like failed to demonstrate value, so we just give up on the project, right? So I hope is like we can avoid at least all 3 of them for sure, by using some of the techniques that we're talking about. So one of the big things that happened in the last decade, I would say, is around deep learning in this space there was a big challenge called Tox21 that National Institute of Health put out this challenge and said, hey, we're going to give you a data set. If you can figure out how to predict the toxicity of these compounds, and we'll have a challenge. Whoever wins will get a prize or whatever. And they did a crowdsourcing of can we actually do this? If we give data, can we actually predict things about the compounds in a way that's actually going to be useful? And they ran this challenge, and turns out there was a winner, and it was actually pretty successful. And the winner actually was called a deep tox. You can look up this paper. It's an amazing paper. If you have a chance to read it. They talk about how they do all the things we just talked about. Like, hey, how do you take a data set, normalize it, take fingerprints, do all these different techniques, compute these features, both static and dynamic features. And they did the ECFP thing we just saw. They did 2D and 3D features. And then they start training models, SVMs, deep learning models, random forest, and all the other things. And in the paper, they say the deep learning actually does much better than any other algorithm. And then they do cross-validation and whatnot, right? And it outperforms everything else. And the cool thing is that in this particular challenge, they were asked to actually predict multiple tasks. It was not just one output or variable. It was like over 10. And they found by doing this thing called multitask learning that the deep learning network was actually able to learn all of these things at the same time and get better, which sounds crazy, but it actually seems like it worked. And of course, the reason they also say is it actually works is because these 10 different things are sort of highly correlated things. When there are 10 different things that they're trying to predict, they were correlated, and there were latent features in the molecules that could actually predict all of these things, even because of the correlation. Just some of the numbers that compared to other models like SVM and so on. All right, so that's sort of the recap. Right now, hopefully you guys I've set the stage, and then we can go into the details of the case study itself that we did with IDRI. I think we have enough time. Okay. So IDRI, just a little background, it's an infectious disease research institute based out of Seattle. They mostly work on vaccines for diseases like Zika and tuberculosis and so on. They have different vaccines and different things at different stages of the pipeline. By now, you should be familiar with all those sort of stages, discovery, preclinical, phase 1, phase 2, and all that. And we've been working with them for over a year now on trying to figure out how we can accelerate their tuberculosis vaccine, the newer version of that they're developing, and speed up their R&D process. And remember that funnel we saw, like where this top of the funnel, there was like lots of things, and then the next stage you want to have hits or leads. So that's the stage we're sort of been working with them on. There's two main use cases we want to focus on. First one was how do we prioritize compounds? So like what they do is they have lots of chemists work for them, and these chemists come up with like different compound ideas. They're like, I'm just going to make up this new structure that looks like this because once again, going back to the key analogy, I already know there is a key that works, so I'm going to come up with something similar to the known key that works. And they just keep coming up with like different variations of the known key. The problem is, in order to coming up with the new key, obviously it takes time, but and they can do it. But testing that the new key they made up actually works is still a lot of time. They have to go and run an assay, put it in the lab, wait potentially days, if not weeks, to see what was the actual result of that. And then they have a whole batch of results. They come back, and they're like, oh, 9,000 of my 10,000 keys didn't work. Okay, now we're going to figure out why it worked, and then go start over. And so this was sort of the normal process they were doing, and we're like, okay, can we automate this? Can we train a model to actually predict, predict the rank of the data set that they came up with, which ones we think are going to have a higher activity versus not. And then the second problem was a related subproblem, which is can we determine the actual value of these compounds, the activation value, right? The inhibition growth factor in this case. How much can it inhibit the growth of the tuberculosis sort of bacteria, right? And so the higher the better. Anyway, so for this, we had a training set. This is a publicly available training set. The ML-SMR was mostly smells, the long ASCII-looking thing we saw. The original one had 200,000 chemicals or compounds in it. The target was this, tuberculosis, bacteria, that particular strain. It's the most studied strain. And we filtered down to half of it. And immediately we saw the data set was heavily imbalanced. We'll talk about this is a phase, one of the earlier problems in this space you'll run into a lot is the data imbalance is pretty bad. Literally with that data set, we had it was a 10% positive, which means 90% of the data was had no effect at all on TV. So we had, we were okay, we're down sample, up sample, what are we gonna do? And so we were dealing with that. It was a phase 0 sort of thing. The test set we wanted to use was IDRI's internal data. They gave us 80,000 samples. Once again, pretty imbalanced, 6.5%, had a positive impact. And most of the earlier things is just data prep, right? It's hey, can we normalize this? Use things like RDKit to do the feature extraction, do the fingerprinting, and then other features that we want to add to the fingerprints, like property-related features, like the 1D descriptors I was talking about, weights and solubility and things like that. So that was sort of what we're doing. Okay, at this point, maybe I'll jump through the demo of a simple data pipeline. Oh, I have a pipeline that we built. Let's see if I can do this. Oops. All right. Okay. So this is, hopefully I'm still logged in. It doesn't log me out. Okay. So this is like our the platform that we built. So it has a way you can actually use this to like one, keep track of all your data sets, and also what we call executables, which are just a fancy way of saying like any scripts or like binaries that you're using. One of the things we noticed in this space is that people had like a lot of existing sort of tools that they're used to, and so so we wanted to support where we can take your existing tools and sort of like wrap them as executables. Once you actually do this, then you can start building what's known as workflows. And workflows are effectively a way to sort of orchestrate different executables or potentially even notebooks to be able to do complex things like this. So this is a simple workflow, not simple, but it's a workflow that we built for IDRI. So what all it does is takes this MLSMR data set and also the IDRI data set, and then like it actually like downsamples it, upsamples the positive ones, downsamples the negatives, and sort of like creates sort of a NumPy array with featureizes the whole thing, creates the fingerprint, adds the properties, and then sort of publishes that sort of NumPy array as another data set that then can be used for like modeling. So and then you can like you can just like click run, and then it'll just go run this, and then like runs on the cloud and all that stuff, and you can see like see outputs and stuff like that, and then you can like build like a new, like you can add like all the data sets and execute drag and drop stuff, and I'm not going to do that, but then start connecting things obviously. Anyway, so yeah, I just kicked off a run, but you can also do things like you can look at like outputs, and like it'll show you like, oh hey, here's the distribution of this data set, and like this is how it looks like, and the distribution of the other data set. Like obviously one of the things I'll talk about this was the two data sets we were using, the train and the test, were so different. That was one of the big problems we ran into. But anyway, so that's sort of an example sort of data prep sort of pipeline we built, and then you also have like ways you can actually do projects, which is just in a way sort of grouping different data sets and workflows and things you have for your the project itself. Is one of the things we're doing data science projects, we already like know this problem where like I have like different notebooks spread all over the place and data sets and here and there, and like and you're like you don't know how to keep track of this, and we get into like naming conventions and other weird ways I've seen data scientists sort of keep track of stuff, and we're like, hey, there has to be an easier way to solve this. And so you can just link all of this into one sort of project, if you will, and then export that out. Anyway, so and then you can also publish models and do all that stuff. Anyway, so that's sort of an example pipeline, if you will, that we built. All right. And so that's sort of phase one. We did a cross-fold validation, k-fold, and we saw like, hey, it was great within that test data, trained data set. The numbers were reasonable. It was not great, but the thing we noticed immediately when we started running with the test data set that it was not transferring at all. Like, you see the IDRI testing set, the accuracy is just like goes way below. The recall is not great, and the precision is like horrible, right? And so we're like, what is happening? Why is not this transferring out? And so that was the first big problem we had. And in order to solve that, we came across this paper, and we said, okay, we're going to try this paper, and it talks about how to penalize in order ranking based thing. But unfortunately, the approach really didn't work. But we just exposed a different problem where we found out the data sets were actually so different that the model we trained was not transferring at all, and it was doing barely better than random. So this is when we sort of switched our approach, and we said, okay, maybe if we train a classifier to say predict which compounds are in the top 10%, and then maybe have a regressor that can predict the actual inhibition score, and then train on MLSMR, and then test on the IDRI data set. It's sort of we've considered different metrics and different ranking metrics, and the results were okay. They were sort of marginally better. It was nothing. Once again, the problem was still there. Like if you look at the recall, it's still it was still okay. Precision actually still pretty bad. And so that's when we're like, okay, how different are these data sets? Is there a way to measure how different these two data sets are? And so we started spending more time on and trying to see if it was easier to just actually do that. And so we trained a classifier to actually predict to see if we can actually separate these two data sets. And that classifier actually did pretty well. It was easily able to separate these two data sets and then saying, okay, this is actually the IDRI data set, this is the other one, which told us that there's the distribution, the underlying distribution that was used to generate these chemical compounds were actually different, right? And so that's where we are now, where we're like, hey, transferability is a bigger goal, right? It's a much bigger goal, especially when, and this goes back to the earlier question that had like, hey, like if I have two different chemicals and they have similar like fingerprints, you know, does that mean those chemicals are like have structurally the activity is also going to be similar? And turns out the answer is no, right? And so one of the things we are sort of like a secondary goal now we've set for ourselves is like, okay, maybe transferability is too high of a goal for us. Can we at least detect that the model that we've trained is actually like so far different from the chemical it's actually being tested on? And then can we figure out by data, you know, the similarity metric, some way to actually predict the transferability of the model itself. So that's where we are, and we're actually starting to deploy the model that was, you know, that's trained on like the IDRI data sets itself, and they're actually fine with that. For them, transferability is not a big concern. They're like, hey, as long as we can train multiple models, and like with a pipeline like this, we can like point at a different data set and just train the model. As long as we can train, you know, multiple models for different data sets, they're sort of actually okay with it. But internal goal for us is, you know, is still like, hey, can we tell when the model is actually out of its like sort of optimal working conditions? Right? That's sort of the goal that we're trying to pursue at this point. Anyway, so that was, I have some references to tools and databases we used. RDKit will come up. DeepChem is a great library for those of you who don't know. It sort of like takes RDKit. RDKit is sort of the foundational library that does all these sort of fingerprints and all the other things that you need. DeepChem sort of builds on top of that and gives you sort of deep learning algorithms on top of RDKit. And there's like a bunch of like publicly available data sets, Chembl, you know, PDBind, a whole bunch of other things. It's really useful. CDD, you know, if you heard of that tool, has also a lot of hosted like publicly available data sets. I also have a bunch of resources and papers for this. I think I'll put up the slides obviously up, you know, online, and you guys will have access to this. In summary, like I said, a lot of pressure on the pharma industry to sort of, you know, accelerate time and reduce the cost. Obviously, AI deep learning techniques, you know, show a lot of promise, but you know, the race is on. Everyone's, you know, there's over 100 startups that are trying to actually solve this particular problem in different areas, different parts of the stages of the pipeline, if you will. Still a lot of gotchas, right? And as you say, transferability is a huge concern. Data imbalance is a huge concern. And you know, you can't, you know, if you go in without obviously addressing some of these things, you can get burned. Yeah. That was my talk. At this point, you know, we'll take questions. Thanks, you guys have been a great audience. Yes.
Hi. I wanted to ask if you've considered generative models to help chemists come up with new drugs instead of them manually designing them?
Yeah, no, absolutely. We definitely brought it up, and they were like super interested. So we're actually going to do that. Like, you know, any GAN, as you know, still needs a good discriminator. So we're still trying to solve the discrimination, you know, like the classifying problem. And of course, we're going to try to do GANs next. Yeah, no, that's definitely something we are actively considering and working with them. Yeah. I didn't talk about GANs, but like everyone knows about GANs, right? Like adversarial networks, generative adversarial networks. It's like where you have one part of the network that generates a compound, let's say, and the other one tries to actually predict if it actually have an impact, or and then they play this game with each other until the predictor can't tell the difference.
Right. I think we can take one more question.
Hey, my question is, can you comment on the accuracy from this machine learning model compared to like the classical quantum mechanical approach?
We didn't do a comparative study of those two approaches. I mean, we do have accuracy numbers overall, like our approach did, but I don't think we compared with the quantum mechanic approach. Yeah, it'll be interesting. It'll be interesting to see like what the actual is. I don't know if there are any papers that actually talk about it. Yeah.